The renin-angiotensin-aldosterone system, or RAAS, is a hormonal system that controls blood pressure. It is triggered when there is a drop in blood pressure. This is detected when there is reduced perfusion or passage of blood in the afferent arteriole in the kidneys. Prorenin, produced from the juxtaglomerular cells of the kidney, is cleaved to form renin, which is released into the blood. Renin converts a plasma protein called angiotensinogen from the liver into angiotensin 1. Angiotensin converting enzyme from the lungs converts angiotensin 1 to angiotensin 2. Angiotensin 2 has many effects. Angiotensin 2 is a potent vasoconstrictor and causes vasoconstriction in the systemic circulation, resulting in increased blood pressure. It promotes sodium reabsorption in the proximal convoluted tubules of the kidney, resulting in increased volume of blood, thus increasing blood pressure. Angiotensin 2 also stimulates the adrenal cortex to secrete aldosterone. Aldosterone acts on the kidneys to increase sodium reabsorption. As a result, urine volume decreases and blood volume increases. In the central nervous system, angiotensin 2 has several effects. It acts on the hypothalamus to stimulate thirst and encourage water intake. It induces the posterior pituitary to release antidiuretic hormone, which promotes water retention by the kidneys. It reduces the sensitivity of the baroreceptor response to increased blood pressure so that this response would not counteract the effect of RAAS. All these actions lead to an increase in blood pressure. Now, RAAS system is regulated by a hormone called atrial natriuretic peptide, or ANP, from the cardiac muscles of the heart, which inhibits the release of renin when there is increased perfusion or passage of blood in the juxtaglomerular cells, indicating that the blood pressure has risen and the RAAS system has done its job. Now, we are going to talk about the pancreatic endocrine hormones. This is the insulin, glucagon, and somatostatin. This response is triggered by two stimuli, low blood sugar and high blood sugar. In the first case, there is a high sugar level or high glucose level in the blood. This generally happens after having a meal rich in carbohydrate. This rise in the blood glucose level promotes the insulin release in the beta cells of the pancreas into the blood. After the release of this insulin from the beta cells of the pancreas, this insulin stimulates glycogen formation in the liver. Liver cells take up the excess of glucose and then convert this glucose and store it in the form of glycogen. This process of conversion of glucose into glycogen is called glycogenesis. This process lowers the blood glucose level to the normal level. Insulin also stimulates glucose uptake from blood into the different types of insulin-dependent body tissue. This also lowers blood sugar to the normal level. When the blood sugar is lowered or decreases to the normal level, this inhibits the further release of insulin from the pancreas. In the second case, there is low glucose level in our body. This happens particularly when there is gap between meals or fasting. The alpha cells of the pancreas detect this stimuli and releases glucagon in the blood. After the release of glucagon into the blood, glucagon stimulates breakdown of glycogen to glucose in the liver. Then the liver releases the glucose in the blood. After the release of glucose from the liver into the blood glucose level rises up to the normal level. This glucose is utilized by the different tissues to fulfill the need of energy. When the blood glucose level raises to the normal level or increases, it inhibits the further release of more glucagon. In addition to factors inhibiting insulin and glucagon, somatostatin, released by the delta cells in the pancreas, also inhibits the secretion of these hormones, insulin and glucagon. And that is how the pancreatic endocrine hormones work. We are now going to talk about the growth hormone. The secretion of growth hormone is controlled by two neurohormones released from the hypothalamus. The growth hormone releasing hormone, or GHRH, which stimulates growth hormone secretion, and the growth hormone inhibiting hormone, or GHIH, which inhibits growth hormone secretion. Stress and decreased blood glucose levels trigger the hypothalamus to increase the release of growth hormone releasing hormone and decrease the release of growth hormone inhibiting hormone. Growth hormone releasing hormone travel through the hypothalamo-hypophyseal portal system to the anterior pituitary. Increased growth hormone releasing hormone and reduced growth hormone inhibiting hormone act on the anterior pituitary and result in increased growth hormone secretion. Growth hormone acts on target tissues. First, it increases protein synthesis and decreases protein breakdown. Second, it increases tissue growth. And third, it increases lipid breakdown. Fourth, it increases glucose synthesis and reduces glucose usage. And it increases the production of a number of polypeptides, primarily the liver but also skeletal muscle and other tissues. This polypeptide, which is somatomedins and increasing growth hormone, have a negative feedback effect on the hypothalamus, resulting in decreased growth hormone releasing hormone, decreased growth hormone releasing hormone, and increased growth hormone inhibiting hormone release, therefore decreasing growth hormone secretion. Those are the actions of growth hormone and how it is regulated.